Actually, I wanted to ask, the first thing I wanted to ask was whether you could provide a bit more context on a line you have on your Twitter profile, which says science that causes limited enthusiasm. I think an anonymous reviewer wrote that. So I'm curious, what was the context of the limited enthusiasm?
So this was, it was for a paper, the paper that came out in 2022 from Leonard Wittkuhn in Nature Communications, and I think which has received quite well, and we have worked hard on this, like on all of the other papers. But of course, it had a history of being rejected before. And one of the comments from one of the reviewers I found just quite snide, who said he has very limited enthusiasm, I think, for my work in general, or something like that. It sounded like this person just said, I don't like that person's work. And he said, he didn't say, I don't like the work. He said, I have limited enthusiasm for it. So I'm guessing it was a Brit, but yeah, you never know. I found it funny, so I was like, okay, that's my line of work.
How did Leonard is a PhD student, right?
Yes.
How did he take that as like one of his first papers? I mean, it's because it seems like it's, I don't know, I've gotten very mixed reviews for papers so far, and especially like the first time you get something negative, it's not fantastic to hear. And that seems like it was quite a unnecessarily snarky comment.
Yeah, exactly. I think he took it well. I mean, I tell all the students beforehand, so it's it might get rocky. You might sort of read things that feel a bit unpleasant, but it's totally fine. In the end, it will get published somewhere, and it happens to the best papers. And there's tons of stories of papers that are absolutely fantastic and had before really hard reviews and difficult stories of getting published. So I don't know. He didn't, if he had any kind of strong feelings about that, he didn't let me know. He seemed okay by my calming words about all of this is normal.
Yeah, it's kind of funny, like the long history of very influential papers that were rejected because they were boring or not relevant or had no potential or whatever. It's kind of, and I guess it's just in general, it's difficult to judge, predict the future, I guess.
Yeah, I don't know how true that is, but maybe you have also heard about like Jeff Hinton making the rounds now in podcasts and in different newspapers in this context of AI safety and so forth. But he's also been asked a ton of times to tell his own story. And one of the things that he keeps saying is that initially everyone thought he's completely crazy to believe in neural networks. And that was he said everyone thought it's a stupid idea to work on that when he started working on that. So it was an interesting reminder, if that is true, that, things can turn out quite differently than they seem in the beginning.
So anytime, even if they have legitimate criticism of my papers, I can think, well,
yeah,
you'll see.
Of course, there's survivorship bias, right? I mean, now we hear about Jeff Hinton, who's been so successful, but there might be a ton of others who...
Yeah, I mean, you don't tell the stories about a paper that people criticized and it was just bad. It's just not a great story.
I think they don't get invited to podcasts or something like that.
Anyway, so your not particularly enthusiastic research. I guess we'll be talking about that now for the next hour or so.
Exactly.
I actually had a question, like a general question first. Like, kind of, what's your main interest? Is it decision making, learning, cognitive maps, OFC hippocampus? Like, is it like one thing, or is it kind of just like a bunch of things you're interested in?
I would say all of them, if that's an acceptable answer. I mean, they're deeply related. I started out being interested in learning, and then understood at some point how deeply related it is to decision making. Because decisions, of course, there's a particular process of just reasoning in the moment of the decision and maybe using heuristics and so forth that is not really related to learning or using particular experiences about how that choice that you're about to make has borne out in the past. But in many cases, I guess we get to make decisions over and over again, so we learn from our mistakes, and that's the kind of thing that I'm interested in. And the hippocampus and the orbitofrontal cortex, and a bit of learning and a bit of decision making. I think all of these play a role in that process.
Okay, so all of the above, yeah. I think the papers of yours we'll focus most on today are the 2016 Neuron and the 2019 Science paper. If I'm not mistaken, I think both of those use a very similar fMRI approach and methods approach. So I thought maybe it might make sense to kind of talk about that a little bit before, so we don't get, so we don't have to talk too long about the methods while discussing the content of the paper. So maybe kind of before we start talking about those two papers, kind of what's multivariate decoding? Maybe as a first question, like what is it? How does it work?
So in all of these papers, I think the backbone is that you basically look at your experiment, and you usually have something like different conditions, which we'll get to in a second. And you ask a question that is kind of like, if I have a particular condition, do I see a stable pattern of activation across some voxels in a given ROI or searchlight or something like that? And if that pattern that is elicited by this condition is stable, that means you can decode it. It means you can take part of the data, you train an algorithm to learn what that pattern is, and then the algorithm is applied to new data that it hasn't seen before. And if the same pattern occurs, it's going to say, I think that's this particular condition. So you're trying to predict the condition from the patterns observed. Yeah, and the main difference is that it's multivariate, meaning you're not just trying to leverage information of a single voxel, but actually of many voxels at a time and how they respond to differences in conditions or cognitive processes. And the other difference is actually a little bit more subtle, and some people have criticized it as a weakness or like a particular statistical tweak that you need to be careful about, which is that the analysis is individual. So you train a classifier for every single subject, and the particular association between condition A and the pattern in one brain area can be entirely different in subject A than it is in subject B or subject C. As long as it's stable within that one subject, as long as the same condition always causes the same pattern. And what you average across subjects is only something that presents evidence for stability of a relationship between the pattern and the condition. But the actual patterns can go completely in different directions. They can, like I said, have different directions. Or in one subject, it's only a few voxels that drive the classifier. In another subject, it's all of the voxels that drive the classifier a little bit, and so forth. And that's very different from the univariate approach, where you're actually asking a question about, is it true that in all subjects, a particular voxel reacts in the same direction, goes up, for instance, when people, exert more mental effort when they are in this one condition compared to the other. So you allow more flexibility about inter-individual differences in the precise relationship between the brain and the condition.
You mentioned that it can be very different. From the studies you've done, is it very different across participants, the activation pattern, or is that more like kind of theoretical concern? So you mentioned some examples earlier. Is that something that can happen, or does it actually happen very much like that?
I think it depends on the brain area, and I would assume that it happens more in higher order brain areas like the orbitofrontal cortex than in maybe primary sensory areas that have a more homogeneous organization also across populations even. So I would think that, yes, that is generally true. But actually, now that I think about it, it's probably also different between different signals. So when you look at value signals in the orbitofrontal cortex, for instance, or the VMPFC, there is some understanding about a consistency of the sign of the response with increasing values. So the imaging signal would usually go up with increasing value or value difference. And that's true for all of the participants. But if you have something like states, I'm not sure that the patterns of voxels activated for state 1 is the same across participants. And in the hippocampus, I would also guess it's tricky. But I think Mariam Ali has some work on the link between the multivariate effects and the univariate effects. And I think she says that it's weak in the hippocampus. So in the hippocampus, you really kind of get different answers from the two different methods, more so than in more cortical areas. But there's probably a lot of research that I'm not aware in this.
Yeah. By the way, can you send me a paper by Mariam Ali that does that? Then I'll put it. For people, and I always put the links and references in the description of the podcast. So I'll put that there in case anyone's interested.
Absolutely.
So you said that the potentially quite large differences between people, that's been a potential criticism of the method. Is it a problem, or is it, what's a feature or bug, that kind of thing? Can't that be? I don't know. It sounds like it could also be, very beneficial that you
I think it is very beneficial because it does make sense, particularly in higher brain areas, that the kinds of responses that you see are learned and shaped through the individual histories of participants rather than some kind of evolutionary shaped structure of the visual pathways and the neurons, how they react to particular visual quantities and so forth. So I think it makes sense. But the criticism that some people have expressed is that it also increases the danger that you are just picking up on a bunch of confounds. So in one participant, it might be that condition A is different from B because that participant finds condition A more effortful. So there's more activity in this direction. And in another participant, they find the stimulus more attractive in condition B or whatever. So it can be, the claim was there could be a bunch of processes subsumed by this predictiveness relationship that you're extracting with decoding methods, which is true, but you can control for that. And I think the benefits are also quite
clear. How can you control for that? It sounds like to me, like when you said that, it sounded to me like that's basically that kind of method is someone that you can't control for because you don't know specifically, if someone's like, oh, I really like orange, therefore they, remember that state or whatever.
Yeah. Well, control if you have the variables, right? If you have reaction times, for instance, I think there was a series of papers from Todd and others who basically said reaction times are one particular concern, and you always have to make sure that's true. But there could also be attentional differences and so forth. But yes, if you can't measure it, then you can't control for it.
Okay, so what kind of, I hope this is not too broad, but like what kind of study or question maybe more does this method lend it to? I mean, it seems to me like you need a particular kind of data data structure that you're interested in. Yeah.
I think you can actually use it for every study, and I think it makes sense to use it for every study. It might, in some cases, pick up on the very same thing that a univariate analysis would pick up, and in other cases, it will pick up on something different. That's the danger of just using it maybe without ever looking at univariate data. And as I said, it could actually have sensitivity to different kinds of signals. There might be part of the signals that are pretty similar across participants, and they elicit the same kind of magnitude response in a particular brain area. And there might be other bits of the response in the brain that are a bit more idiosyncratic, but nevertheless meaningful. And I think there are even people who will basically try and really separate those out and treat them as different quantities. You can subtract the mean activity pattern across conditions and then basically just try to decode from the residuals, if that makes sense. And it's not necessarily said that one is better than the other. There are probably cases of both where you find, for instance, relationships to behavior with decoding scores, but not univariate activity, and the other way around. I wouldn't be surprised if that is true. These are probably just different aspects of the activity that is going on during our experiments, as far as we can see it with fMRI.
Yeah, okay, so I guess that was a kind of a rough introduction to the topic. Maybe we can be a bit more specific now, kind of like, not necessarily like specifically how you use that method, but I guess we can talk more about what you actually did, and then it maybe becomes a bit more concrete. So yeah, your 2016 paper, Human Orbitofrontal Cortex, represents a cognitive map of state space. Basically, what were you trying to do? What did you do? And what did you find? As like mini abstract.
So maybe I'll start with a little bit of a backstory, which started before I joined Yael Neff's lab in Princeton. I think Jeff Schönbaum basically approached Yael and said that they had some recordings from dopamine neurons in rodents in an experiment where they had lesioned the orbitofrontal cortex, and they couldn't really make sense of them. And it looked like as if the prediction error signals in the dopamine neurons, they were altered, but in a quite subtle way. They were altered in some conditions, but not in others. But that alteration seemed reliable and not random, but it wasn't clear why is it this condition and not that other condition. And then Yael sat down with Bob Wilson, and in a 2014 paper that came out, they basically proposed the solution that what happened in that experiment, in that data, is that the animal was confused during some stages of the experiment, which stage of the experiment it was in. It just wasn't sure, and therefore it had kind of blurry predictions about the reward to be expected, and that was the result that they then saw in the prediction error differences. So why was the animal confused? The argument was that the lesion of the orbitofrontal cortex had kind of impaired the animal's ability to distinguish between stages of the task that are difficult to keep apart when you only rely on sensory information. And that's what's called partially observable task states. It's a bit of a technical term coming from the RL literature. And the idea is basically, sometimes it's quite easy. There's the green light and the red light, and it's very clear, okay, these are different situations. I maybe have different reward expectations when the light is green and when the light is red, or there's different odors when I'm this little rodent in the experiment. But sometimes things get a bit more complicated in the sense, for instance, that it could be that the rat had waited a long period before receiving the reward in the last trial, and that means that in the current trial, it only had to wait a short period. So knowing that now it's in the stage where it only has to wait a short period before the kind of drop of water comes is something that is not as easily detectable from the environment in the moment. You have to use your memory, you have to kind of accumulate over time or have a sense for time, which is also something that is not easy to observe. And the idea that animals lost that ability to distinguish between these more subtle task stages that are not directly signaled was basically the solution to explaining the differences in the prediction error. So what we did in the paper that you asked about is when I then joined Yael's lab was that we asked, are those representations indeed in the orbitofrontal cortex? Does the orbitofrontal cortex indeed distinguish between different states of the task, in particular when those states of the task are not signaled with any kind of sensory cue in the experiment? And so what we did is that we designed a task in which people had to make decisions about stimuli that were kind of ambiguous, and the kind of decision they had to make didn't really depend on what exactly is on the screen, but on the history of responses that they had given in the previous trials. So it was a task in which you had to pay close attention, and then basically on every trial integrate information from the past in order to know, okay, I'm seeing this particular stimulus, and I know that now for me it's time to pay attention to this particular aspect of the task and give this response. And maybe more concretely, what happened is that people were looking at pictures of faces in houses. They were semi-transparent, so there was always a face on the screen in a house, and they were overlaid. And we told them initially that we would give them a cue for the first trial that would tell them, okay, you have to start looking at the faces, and you tell us in every trial whether that face is old or young that you see. But once you notice that there's a change in the age of the faces, so you might see the first age that's young, and the next face is also young, and then the third face might be old, that's a signal for you that now you have to pay attention to the houses on the next trial. But there's no cue in the task. It was just you knowing that beforehand you had paid attention to the faces, and they were young faces for a bunch of time, and then they changed, and that was the signal for you to switch your attention to houses. And then you would continue to see the same stimuli. They weren't any different, but now you would know, okay, I have to look at the houses and do a similar old-young judgment in the sense of, is this house modern or more non-modern architecture? So people were doing that task and basically always giving us a response old or young, but in their brain, they started to make this more subtle distinctions between which task state am I in. Do I have to pay attention to the houses? Did I just notice a switch and maybe on the next trial I have to switch my attention? And these kinds of things. And that's what we tried to decode from the orbitofrontal cortex, then asking the question, does the orbitofrontal cortex provide that distinction between the conditions? As Yael and Bob had basically hypothesized based on their interpretation of the data that I mentioned before. And that was the case, pleasantly to our surprise. It worked out, and when we looked at the different pieces of information that were necessary from the task, they were all there in the orbitofrontal cortex. And we had started out initially with just looking in the OFC and making sure that this is where we can decode. But even when we started to look across the entire brain, we found that it was pretty uniquely the orbitofrontal cortex, a medial part of the orbitofrontal cortex, where we could find those distinctions between partially observable task states.
And luckily that fit with Schoenbaum.
Exactly. Luckily that fit. Otherwise, I might have lost my postdoc position. I was not like that, but that was a good start, I guess.
Yeah, I mean, I'd like to ask a little bit about the task, because it seems like a slightly weird and artificial task. And it seems to me that this is, for many tasks, that's quite the norm, right? You have a task that if you're the first time you're doing that kind of task, you're like, what am I supposed to do? This is super weird. Like seeing these superimposed images of houses and faces and then deciding between them and that kind of stuff. Maybe as a first start, like why in this particular experiment? I mean, Houses and Faces has obviously a long history in cognitive neuroscience, but why did you take that kind of approach to this question?
Yeah, so Houses and Faces, that was basically a plan B. That was just in case we can't find anything in higher order areas. There's something of a, they have been the workhorse of cognitive neuroscience for a while, because you get very reliable responses to those in inferior temporal cortex, in the parahippocampus, in the fusiform face area. And we just wanted to make sure that we have something to look at in case the orbitofrontal cortex analysis doesn't come out. And I didn't end up looking at them. Well, you can clearly see when you just ask the question, can you decode whether it's a face or a house that people are paying attention to, then you end up in these brain areas. But these are not the same brain areas where you can decode other information that's relevant for the task stage, such as whether it was young or old. That's just somewhere else, because it's visual in nature. So it was basically pragmatics. It wasn't anything specific about faces and houses. They were overlaid and semi-transparent, and they looked that weird just because we wanted to get rid of any confounds in terms of eye movements and so forth, and wanted to make it really hard to basically just find any kind of shortcut to do the task based on purely visual information. And of course, then you can also, with that setup, you can perfectly control the frequency of switches, the frequency of trials in which people pay attention to houses, the frequency of old images and young images, and the transition probabilities between the images that people are not paying attention to. I'm that kind of person who gets really antsy about these kinds of things to make sure there are no confounds. But it's really just due to that kind of knack for detail and worry about confounds that the task ended up being so artificial. I should say, though, two things. So the one thing is that I think the topic in general is not, in my mind, this is totally not some kind of artificial phenomenon that only occurs under rare lab conditions. But I think it's really fundamental. I think the kinds of information, we have to generalize all the time. We have to all the time make a decision about when something is rewarding or not rewarding, or a correct response or not correct response. What does that mean for all of the other things that look similar or maybe not similar to what I just got the reward for? And so you have to ask yourself, which are the things that are similar and which are the things that are not similar in terms of, are they giving me the same reward when I respond to them concretely? And that is not easy, even in the case that we kind of made with this experiment, in the sense that sometimes two things that look completely identical can give you very different responses. Very simple example would be, there's an amazing coffee shop and you can go in there and you can have an amazing cup of coffee, and that's wonderful. But if you had 5 cups of coffee on that day, if you go in that very same coffee shop, then you will get pretty bad heartburn. So it's not a smart idea. And I think that we have to kind of contextualize and de-alias sensory information all the time in order to make our decisions in real life. And the task is meant to reflect that. So that's, I think, one aspect. The other aspect is that I have an interest in making tasks more ecologically valid. And we have run one experiment now that we hopefully will publish at some point. We're still in the analysis stage, in which people play more of a video game. So there's many more degrees of freedom. They can walk around freely across different rooms. The kinds of rules that they learn are more complex than the ones that we used before. And it feels a lot more natural. It feels like an 80s computer game. And the question is very similar to what I mentioned before, in terms of, does the orbitofrontal cortex help people to differentiate between situations that look quite similar but are actually meaningfully different considering the structure of the task? And we find the same thing even under those circumstances, but we have a lot harder time in our analysis because we need to control for all the visual flow variables and so forth that come with the computer game setup.
Yeah, I know it's kind of a, how should I put it? I mean, I guess it's just a very difficult topic in general, that you want to, have a task that's as precisely controlled as you can be, whilst it also being natural. And to some extent, it seems to me, I mean, they are not exactly antitheses or like pulling in different directions, but they kind of are, they're, let's say, orthogonal to each other at the very least. Yeah, I mean, I guess I'm very aware of like how difficult it is to have a nice controlled design and everything, but yeah, I do often, particularly with kind of decision-making tasks, wonder like, because I agree that the what it represents and what you're measuring with it is fundamental, but then you always feel like, yeah, but can't there be like a more natural way of testing this if it is so fundamental? And I mean, this is not, specifically about your task. This is just something I've had a few times. I mean, I know with a two-step task, I once took part as a participant in that, and it took me a while to understand it. And apparently lots of participants just don't really get it, or it takes them ages. And yeah, I mean, I guess it's just like a difficulty of the field. But it seems to me like, particularly in kind of decision making that comes from like the reinforcement learning side, there's a lot of very weird tasks that take a long time to explain.
Absolutely. I mean, maybe I think one way of looking at it is that it's undoubtedly true that you, when you do these artificial tasks, that you get a very precisely defined or a more precisely defined experimental situation and a more precisely defined interpretation of your data, because you have ruled out a bunch of other factors that could have influenced your data. But the danger is, of course, that the truth of that finding might be very narrow, in some cases even narrowed down to that very precise setting that you set up in your experiment. And in the case of this two-step task, as soon as you change even something minute like the instructions or what you emphasize in the instructions, or maybe the stimuli and so forth, then because things interact and humans are complex, and of course they're very sensitive and pick up on all of these things, then behavior might change and cognition might change. So you might have sort of found something that is true, but only true in that very sort of limited circumstance. I think, the interpretability of the data is cleaner, but the generalizability there is, of course, you need to run the same task with a different setting where you change a bunch of variables in order to understand, is this really a general phenomenon, or is that something that only happens in this particular situation that I forced my undergrad participants into?
But I guess it's also a situation where you kind of not the only way is through, but like you have to start somewhere and figure it out. And as you said, with the houses and faces, there's a good other reasons for using that. So why not? Yeah, maybe. I mean, we haven't actually talked about the findings that much, but I thought maybe actually we could go kind of straight to the science paper, because I guess to some extent it seems to me that you were like, well, that worked. Let's do it again, but slightly different. Looking at like one step further.
Yeah, so there is actually the conversation we had just now is interesting about that paper, because there I should say that wasn't the plan with the paper, what came out of it. That was more of a like accidental finding, if you want. But maybe first off, so what we did in that paper was pretty much the same that we did in the paper that described before. So people were doing this weird task with the faces and the houses and old and young, and they had to switch, and there were only I guess two changes. One was that participants learned how to do the task inside the scanner. We even instructed them inside the scanner. And then they did it over two different sessions, over two different scanning days. And the second one was that we asked participants to rest in the scanner before and after each task session, where their instructions were just that they can take a rest, lie in the scanner, keep their eyes open, and the usual resting state scanning procedures. So no particular instructions what to do during these periods. And so what we had originally wanted to do is to look at how these state representations emerge in orbitofrontal cortex. And there's a bit about that actually ended up in the paper. But the main finding that I made, and it was literally when I was starting to look at the data, and I have no idea how that idea came about. I might have just seen a talk about replay or something like that. But what I had basically noticed is that I can also decode the task state from the hippocampus, just not as well than in the orbitofrontal cortex. And beforehand, I hadn't really seen that or paid much attention to that, because if you do whole brain correction, for instance, then it doesn't really come out, or maybe it was too close and the orbitofrontal cortex kind of took center stage. But there was a signal in the hippocampus that I believed in. And what I then did is that I looked at the resting state data and asked, well, that classifier that I trained on the task data that would now give me, if I give it a new set of data that it hasn't seen during training, will give me a prediction about which condition it believes the participant is in currently. If I apply that classifier to the rest data, what does it tell me? And it turned out that the classifier started to make predictions whose sequences kind of looked like as if participants were playing the task in their minds while they were lying in the scanner. And of course, it was very subtle. It wasn't something that you could immediately see with your eye, but it was big enough, actually, the effect, that without having a very kind of sophisticated approach at that time, I happened to spot that. I happened to see that, wow, the transition of this task state number 13 to whatever other tasks that usually came, that seems quite frequent. That's the one that happens in the task as well. It's not a random one. And then I started to look at those transition probabilities more systematically. And what came out of that is that we found that apparently during the resting periods after the task, but not before the task, participants show signs of sequential reactivation of those abstract task states that they underwent while they were doing the task. And that was, first of all, we couldn't believe it ourselves. I was skeptical, so was I. We had a bunch of meetings with many people in Princeton back at the time who all found it quite exciting, but kept asking us to kind of do more tests. We got more data at some point, do more sanity checks and so forth. So there was a period also where I faced limited enthusiasm that we mentioned before, but it survived, I guess, all of the kind of tests, and we convinced ourselves that the signal is real. And what is really exciting about it is that there were activation in the hippocampus during resting state sessions that were sequential, that few people had seen in fMRI beforehand. I think people had seen reactivation in the hippocampus at that time, in the hippocampus in the sense of, if people have more experiences with houses than faces, you would see more activity of house-related brain areas during resting state conditions. But you hadn't really seen the kind of the precise sequential structure. And the other thing that was exciting is that these were not observable things that people had seen, but these were the abstract task states. There was not a replay of, okay, the particular picture was followed by that particular picture, was that followed by that particular picture. But it was what participants thought about that picture when they saw it, and the picture that came afterwards in the light of these task rules that required them to think about similar looking pictures quite differently, depending on whether they're looking at the faces and the houses and so forth. So that was quite cool, and then I think paved the way for a bunch of other work that we're now doing that is kind of continuing to use fMRI to look at these sequential signals in the hippocampus and elsewhere in the brain.
Maybe as brief context, can you provide a little bit more of like what replay is? And I guess there's quite a lot of research there, just not in fMRI.
Yeah. So the original finding is that when you record play cells of a rat while the rat walks around in a maze, and you see they are spatially selective and so forth. But when you keep recording them when the animal sleeps, I think the first studies were in sleep, then you find that those cells reactivate sequentially, again, in a manner that looks like as if the rat is kind of mentally walking along those paths that it had walked before. So the sequences are not random. And in the original findings, those activation sequences are actually highly compressed in time. So the kind of typical figure that people refer to is that it looks like as if the animal is walking 20 times as fast in their mind as opposed to what happened in real time. So it would only take 100 milliseconds to walk a path mentally, or see the reactivation associated with a path that took 2 seconds to walk in real life, something like that. And I think starting from that original discovery, people have found that there's a pretty large diversity of that phenomenon. Happens in sleep, but also happens during resting periods. can be forward, sometimes can be backwards. There's instances when it's slower and instances when it's faster. But it became pretty clear also that it's not just a random phenomenon, but it's related to memory consolidation. I think that's the strongest case to be made as of yet, in the sense that reactivating those sequences leads and is associated with stronger memory afterward. Yes, and so what we found in the 2019 paper is kind of first of all, few people had seen something that is much closer to the animal observations in humans. And it's also broadening the phenomenon away from a reactivation of place cells to a reactivation of really abstract representations that happen in a mental space rather than in a physical space.
I was wondering, is that, non-spatial is one of the words in the title, non-spatial task states. I mean, was that part of the special thing that it wasn't an animal, or human or anyone like walking around an arena and you being able to trace that back, but like a very, as you said, highly complex and abstracted from the sensory information.
Yeah. I think, part of the reason why we wanted to emphasize that, because there is maybe perhaps until today a discussion ongoing, as you might know, about what is the nature of those cognitive maps in the hippocampus. And the cognitive maps in the hippocampus originally have been observed in the context of spatial navigation experiments, coming back maybe to the discussion about the specific tasks and the culture of tasks that we have in using. And it's just true that in the rodent literature, people use spatial tasks all the time. They are really well trained to find cells that are spatially selective, record from them, and then do all sorts of tests from them. So the picture that had emerged is this is a spatial brain area, and replay therefore looked like it's a reactivation of spatial trajectories. But that, I think, strong formulation started to crumble at the time, or was already kind of questioned by many, and many people said that the cognitive map is much broader, can refer to non-spatial aspects. And the fact that replay, which is kind of a mental walking in your kind of mental map, happens about these non-spatial abstract task states, or involves these non-spatial task states, was kind of further adding to that discussion. I think that's what we were hoping to say.
Yeah, I had one other question about, this is, you mentioned this quite a bit in the paper also, and I think it's, because it's worth mentioning. The question is like, how can you, I think one thing that seems so, at least to me, so incredible about the paper is that you're using a very supposedly slow method that, averages a large space of time to get at something that happens very, very quickly and very, not quickly, but briefly also. So I don't know exactly where I'm going with this, but can you maybe maybe try to explain, if kind of how can you, it just seems surprising to me if you have like a 1 or 2 second window, over which you're averaging that, like something that's 50 or 80, whatever milliseconds long would have such an effect you could even measure it.
Yeah. No, that's a really good question, actually, one that we kept asking ourselves at the time of the paper, and I continue to ask myself after the paper was out, and we've written about that since then, because it does seem surprising. And I think there are two very simple intuitions that help a lot with that. The one is, yes, the bolt signal is slow, but its kind of slowness helps you to pick up on events that were really, really short. So even if the entire replay sequence happened within 200-300 milliseconds, we have a bunch of seconds in which the signal is going to be influenced by those replay events. In the bolt signal is going to be influenced by that. So we don't need to rush and basically say, okay, we need sequences where we need to sample every 10 milliseconds the bolt data, because whatever consequence a replay event has on the bolt data, whatever signature it kind of kicks off, is going to be in the signal for like 10 to 15 seconds. So that's our chance. The other question is, what kind of response does it cause when you have these like really fast activation sequences in neural activation patterns? And what we are observing is our bolt signals, not neural signals directly. And I think the worry that people had, or the intuition that people had beforehand, including myself, is that if it's the very same voxel that would be triggered by those events, then you have a big problem, because the voxel in response to event 1 starts to rise, and then if the other event comes on and causes the voxel whatever to rise even more, or maybe decrease a little bit, that is very difficult because the interference processes that happen within the voxel and the slow response it has to like one event. But if it would be true that in the ideal case, voxel number 1 responds to event number 1, starts to rise, voxel number 2 responds to event number 2, or at least a little bit more so than voxel 1, and starts to rise a tiny bit later, then you have something you can pick up on. And those are activation differences between the two voxels. And combined with the first fact that I mentioned, namely those activation differences are going to stick around in the signal for 10 seconds or so. That is basically, I think, the fortunate combination that we are picking up on, in the sense that there's different events are carried by different voxels, and they the temporal difference eventually translates itself into a kind of activation difference, and that activation difference signature of replay, if you want, is going to be present in the signal for a couple of seconds. So even when we have a TR of only 2 seconds, we get to see it.
Yeah.
Does that make sense? I mean,
yeah. I mean, I'm curious, like, did that then change like how you design your experiments from now on, because you realize you can pick up on more stuff than you thought you could, or?
Yeah, I think, one thing that we're doing in a lot of experiments now is basically to put in pauses of longer periods where nothing happens. So people are in the task and they do some conditions, and then we just stop it, and there is whatever fixation course on the screen for 15 or 20 seconds. And those 15 or 20 seconds that are clean of any ongoing visual input are the window where we basically can look more precisely at the aftermath of whatever happened in response to the former stimuli that might involve, for instance, spontaneous forward sequences starting from the stimulus you have seen last. I think that's one particular consequence. We have collected data on the question whether it is worthwhile to sample faster. As I mentioned before, so in fMRI, typically the TR, which is the sampling frequency, is about 2 seconds. And I'm not super convinced it will pay off, but we are about to look into that in more detail. And it's possible that this could be an important factor as well.
Yeah, it's really interesting. It's kind of fun how there's just so much more you can do with fMRI than even I was taught in my master's, where it's like lots of neuroscience, and it seems like even since then stuff is regularly coming out where you're like, oh, we could do this also.
Yeah, absolutely. I mean, I should say that even though I work in the field, I kind of made a career out of trying to do things with fMRI that are hard to do. I wasn't quite sure at some point how much of a future it really has, and whether we haven't just reached the ceiling here and we just have to wait for another fundamentally different technology. But, recently, I mean, there is, Alex Hutt's very impressive work on semantic decoding that came out in Nature earlier this year. There are a number of other groups who do really impressive stuff with fMRI. So I think I might have a future now working with the same machine. I don't have to retrain and learn how to work with a new kind of machine that's yet to be invented.
That's nice that you have a future. That's good.
that would be good. I mean, no one knows.
Yeah. I mean, this is a hypothesis.
I live under the illusion that I have a future.
Yeah, you have. Yeah, it's vague optimism.
Exactly.
Yeah, maybe to slightly broaden kind of the conversation, kind of what, taking both of those studies together and like all the stuff we've discussed, and maybe a very simple question. What does orbitofrontal cortex do?
So
I'd say a brief question first. Is orbitofrontal cortex the same as ventromedial or prefrontal cortex? Or is there a difference between the two? Or because I think they seem to be sometimes used kind of synonymously.
So no, they're not the same, but they overlap, and that's where the confusion in the terminology comes from. So orbitofrontal cortex is the kind of the most ventral bits of your frontal cortex that just sits above your eyes, and the thing to know there is it's the whole kind of ventral surface, meaning it goes all the way from the medial parts up to the kind of more lateral parts. And much of the animal work actually focus on the more lateral parts, but it includes a medial part, and the medial part actually stretches a little bit dorsally and is pretty connected to all of the the medial parts that we see more dorsally that would be whatever variously referred to as VMPFC, then going into DMPFC, maybe even ACC and so forth. That's what some people have called, I think, the orbitofrontal medial network or so. So there's a continuity. There is a bit of the medial part that is just part of OFC, classically, anatomically speaking. There is maybe a bit of the VMPFC definition that doesn't exist anatomically, but is more the functional definition that you would say, well, anatomically, that's just outside of the borders of orbitofrontal cortex. But if you look at connectivity patterns, it seems to be functionally the same thing. So, yes, it's a bit confusing. There are other things that we shouldn't get into because they're very confusing about the homologies between rodents and humans in the medial and the lateral parts and so forth. Very complicated. People can get upset about that. I wanted to stick with OFC because that's how we started out, but we actually changed to talk more about VMPFC because we end up finding effects in the medial area, and maybe that is a terminology that's just better understood by the field and speaks to more people than the terminology of orbitofrontal cortex, where what we really mean, at least in terms of our findings, is the medial orbitofrontal cortex. And that's, I should say, at least in humans, because many of the findings from Jeff Schönbaum's lab, who records in rodents, come from lateral OFC. But whether those are not medial now after a bunch of million years of evolution, it's not quite clear.
Yeah, but is it? It seems like there was one quite cool review paper that I think it came out last year by Joni Wallace and someone else. I think I've literally said that sentence before on the podcast. I should look up who this other person is. That I think talked about like the hippocampus OFC connection and kind of this question, like how is it related in terms of like lots of the OFC stuff in rodents is also with value that comes out with economic value and that kind of stuff, and then but also the cognitive map stuff. So it seems like the stuff you do is quite related to that. So I'm just curious, like kind of what's your view on this from having done quite a bit of work on this?
Exactly. So first of all, yes, the connection to the hippocampus of the OFC is actually more strongly so in the medial parts of OFC, as far as I know, at least in primates. So that's kind of yet another reason to maybe why the medial parts of the OFC is where we are finding things. And the medial part of the OFC, or VMPFC, then how it's called net literature, is also where people find values. And I think in combination, my interpretation of what the orbitofrontal cortex does is, of course, it's not just one thing, but it seems to be very clearly related in decision-making, in value-based decision-making in particular. And there is not just one thing that needs to be done, so to speak, computationally, if you want to solve a value-based decision-making task. But there are these kind of at least two different categories. One is you need to store and retrieve values of how basically your expectations, how good is it to do a choice in response to that particular situation or stimulus. And the other one is you need to have a mental map or like a set of states that tell you basically how to sort, keep apart, or merge these different reward experiences that you have across your sensory space, in the sense of you might get reward in response to when you eat some vanilla ice cream in ice cream shop A, and the question is, does that generalize to all the other flavors in that same ice cream shop, or does it just generalize to other vanilla ice creams in other shops? So you need to, when you learn about rewards, you need to know what things are equivalent in terms of how rewarding they are, or how I can apply my current learning experience to those other sort of related or possibly related things. And that's what the cognitive map does. So they are really, really related to each other. It's not just forming expectations, calculating prediction errors, which is what the typical value literature is focused on, but it's also what precisely to update with those experiences and how to generalize. And I think both of these functions are in the orbitofrontal cortex, and they interact with each other. And yes, we don't have time to get into that, but there's a very recent paper from Nia Moneta and me, where we go into that question and look at values and states at the same time in the orbitofrontal cortex. So yes, I think it's basically representing the map and the values, and they are very, very closely linked to each other.
Yeah. Yeah, so I mean, I'm trying now to have some questions that repeat from from one episode to the next. So I'd like to add those kind of recurring questions now at the end of the episode. The first is, do you have any? Do you know of a paper or a book or I don't know, some sort of document of some sort that I don't know, more people should read, or that something that's been forgotten, or I don't know, just a personal favorite paper of yours or whatever, something like that?
Yeah. So there are two books that have really kind of influenced my whole thinking, have been very influential. Two technical books. One is Sutton and Barrow Reinforcement Learning. The other one is the machine learning book by Chris Bishop. I think it was only in combination with my knowledge about neuroscience and psychology that they kind of really helped me to think about some things. But those were like the few books that I have maybe read in such a level of detail in those two books. I would say so. I would recommend them. In terms of a paper, doesn't happen very much, but I happen to have just read one of the kind of old classics, namely this old paper from Shepard about universal laws of generalization. That's from the mid-'80s or something like that. And I think it's a really interesting article. I wouldn't say it's beautifully written. I actually found it sometimes difficult to follow and a bit abstruse. But the ideas behind it, I think, are really, really striking and I think he was ahead of his time, and they are super relevant even today. And I think the major thing that he says there is that instead of looking for kind of regularities in behavior that are universal across some stimulus space, so how do people generalize when you go from frequency 100 hertz to frequency 120 hertz to frequency 130 hertz, we should recognize that people might have different internal mental representations about how similar those frequencies are. And when you have figured that out, then the generalization is actually very irregular. It might be very kind of law-like. That's the claim he makes across different participants. But he places the emphasis on kind of the internal map of people and variability therein on the psychological space, how he calls it. And says when we have understood what the particular psychological space of a person is, within that space, things might start to behave more regularly and even more homogeneously across different participants. And I think that general insight is absolutely important and significant even today as we start looking more at the hippocampus and the orbitofrontal cortex as possible neurosites, understanding metrics that could reflect those mental spaces, grid cells, and so forth. I think that's a cool paper that I would recommend reading.
Sounds very cool. Yeah, I haven't read it. I mean, I've obviously heard the name, but I think I have to read it now. As before, I'll find the references and put them in the description. Just brief question. I guess I don't actually know what your background is, but from the readings you said you liked, it sounds like you didn't study computer science, but you're still very influenced by it now because both of the books are basically computer science, right?
Yeah. I did study a little bit of computer science. So I had a bit of a life crisis at the end of my undergrad in psychology, where I was really unsatisfied with how difficult it was to quantify things. And then I did a 1-year exchange in Toronto to do machine learning. And that was academically an extremely intense year because it was completely out of my depth, sitting with all these computer science students and trying to kind of solve all of these assignments. And it was fortunately enough Jeff Hinton, who was basically whose class I was visiting and doing all the exams. But it was hard, but I survived and it has really influenced me. But I think only in the combination with kind of the neuroscience psychology bits that I had already brought to the table.
Yeah, I know the sense of feeling out of your depth with computer scientists. I once wanted to take unsupervised learning at UC at the Gatsby Center, and I was like, I mean, the thing is, I just didn't know any of the notation, like the mathematical notation of anything. So I was just so lost. I just ended up not taking the course because I was like, I have no idea what any of this means. I think I might fail this course. This is probably. I think there's other cool stuff I could do here. I don't have to do this. But how did you?
Yeah, no, it was difficult. I guess what helped me in this is that I had moved to Canada. I had a whole year ahead of me, and I had only signed up for these like 2 machine learning courses. So it was like, man, I have this whole year ahead of me. And somehow I found it really interesting. And I have to say that was also about Jeff Hinton and his style, because the way he talks or talked in those lectures, and I see that when I see interviews of him today, is very clear. It sounds very simple and incredibly logical, as if it's like the most obvious thing. And he just walks through neural networks and whatever he's talking about in a language we're like, yeah, that makes a lot of sense. And then you see the assignments and it's all math and all coding. And I'm like, what? Where's the relationship between this kind of beautiful, very intuitive description that he has given before and those difficult mathematical derivations? But that motivated me a lot to kind of try and understand that. And then I didn't have much other things to do. So I was,
Yeah, I guess the commitment of moving to another continent helps you.
Yeah, exactly. It was also cold and dark in Toronto for the most part. So
yeah,
good conditions.
That sounds like a pretty cool time. The second question is, I mean, I don't know about you, but I tend to repeat mistakes. And I'm curious, anything you wish you'd learned sooner? This can be whatever you want, academic, private, I don't know.
I think I'm still learning this, but it's to focus my energies. Sometimes involves saying no, and involves drilling down on a particular subject that you are really interested in and just keep at it, rather than to continue to kind of do new things and participate in that and participate in this and so forth. I think there's also, I like kind of being out and about and kind of think about different kinds of topics, but I think it's also good to kind of leverage the things that you have built on before and continue that work. So with the science paper 2019, I'm very glad that we continue to do even like methodological investigations, trying to answer some of the questions that had occurred to us, like how is it even possible to do that signal? And I think it's great that instead of moving on, I was like, okay, let's wait a moment, let's try and do a few studies and figure out how does that work. And I'm really glad I did that, because I think I learned a lot from that.
Okay, cool. If I'm not mistaken, for anyone wondering whether they're starting to hear voices, I think that's your kid in the background, right?
Oh yeah, you can hear that.
Yeah, it's not a problem. And just in case anyone, sometimes I listen to podcasts and you suddenly think like, what's going on? Am I having hallucinations? No, that just happened in real life. You're not hallucinating.
Yeah.
And last question. Any advice for, let's say, someone?
You can hear. Yeah. I should see if I can give him some advice.
Advice for, I don't know, scientists like me who are, in some sense, starting out their career.
Yeah. I think the most important thing for me until today, and I see also a lot in sort of younger people, is to be clear on the big picture before you like dive into an experiment, to make sure you can summarize in few words why is it interesting and what's the question, what is really behind that, before you dive into the details of the experiment and the analysis. Because we spend a lot of time, most of the time I spend with my PhD students and postdocs and in other projects on the details, on designing the experiments, on the logistics, and like there are so many things that need clarification, picking the stimuli and so forth. But it's really important before you start to basically know what you want with that experiment and why you want it. And I think that's the thing I would advise people. Otherwise, I mean, I still think it's exciting times in psychology and neuroscience. I mean, academia can be hard, it's difficult. I think many people have realized that and has problems, but the field is just, it's really cool. It's a cool time, I think, to be in it because things are moving increasingly faster. What's happening in AI is obviously pretty stunning, and it might have consequences for us, how we understand our data, how we understand intelligence in biology and beyond. And I think it's an interesting time to work in that field.
Cool. Well, on that positive note, thank you very much.
Cool. Yeah. Thank you for having me.